Now, your individual genetics, you can't really change those that much, but it is important to notice.
Dr. Rahul Gari, Dr. Julio Fiore, Dr. Fax Mwale, and Dr. Joshua Borsabash. We'd also like to thank the coordinators, Nisha Hyun and Adriana Scambatera, for their involvement in being able to make this happen. So thank you so much. So now I will share the student presenter.
Yeah.
All right, I'm just going to make sure that the sound is on.
Good afternoon. My name is Eliane Hamburger. I'm a master's student in experimental surgery in the laboratories of Doctors Haglund and Rosenzweig. And today I will be talking about targeting senescent cancer cells by using senolytic drugs in order to prevent breast-to-bone metastasis. We hypothesize that by combining senolytics with the standard chemotherapeutic doxorubicin, SASP release, tumor size, and overall outgrowth will be greatly diminished. So what is senescence and why are we targeting this? Well, senescence is characterized by cell cycle arrest, distinct cellular changes in gene expression, metabolism, and chromatin organization, as well as changes in the anti-apoptotic pathway. All of this leads to the increase in tumor progression via the senescence-associated secretory phenotype, also known as SASP, which secretes pro-inflammatory and pro-tumorigenic factors, all leading to chronic inflammation, cell proliferation, cell invasion, as well as increased angiogenesis, which is the blood supply to the primary tumor. And all of this leads to tumor growth and metastasis. Drugs that we will be using. So doxorubicin, a standard chemotherapeutic, is a topoisomerase 2 inhibitor that leads to DNA damage and then apoptosis. The two senolytics we will be using are ovanillin and RG7112. Ovanillin is a natural anti-inflammatory senolytic, whereas RG7112 is a p53 MDM2 inhibitor. These drugs will be used alone and in combination with each other. The significance. Why are we studying this? Why is it worth the project? Well, it's been estimated that 50% of US patients dying annually of solid tumor cancers have bone involvement. Chemotherapeutic treatment causes the induction of senescent cells within and surrounding the solid tumor. And this project may offer a better understanding of cancer cell senescence and its relation to chemo resistance, chemosensitization, enhanced growth, and metastasis. How are we studying this? So first off, we're creating a tumor microenvironment model where we create tumor spheroids with cell lines. And then once we've created the spheroid, we surround it with collagen, as in the matrix, with surrounding cells, i.e. fibroblasts, to test the outgrowth of the tumor and the growth itself of the primary tumor. So what are we going to analyze? With fluorescent imaging, we'll measure the area and the outgrowth. We'll look at test the viability and the activity of the cells with Alamar Blue and live dead assays. And we will collect media to look at the cytokine release and SASP effect. And we will also be staining for the increase of senescence, apoptosis, and proliferation. Preliminary results. So first off, we wanted to show that doxorubicin does in fact induce senescence, and with increased concentration of doxorubicin, you will see an increase of senescence within and surrounding the primary tumor within our model. So here you can see on the left that within the breast cancer cell line, there is an increase in senescence with increased doxorubicin. And then on the right is a fibroblast cell line, where again, you see an increase of senescence with an increase of the standard chemotherapeutic. Now, this is looking at the area and the outgrowth of our spheroids in this model, where you can see that in our model, doxorubicin does in fact reduce spheroid size as well as outgrowth, which means our model is working. Lastly, I wanted to show that viability and area is reduced greatly, and even more so significantly, when all drugs are used in combination rather than alone, and in comparison to doxorubicin alone or the control. In conclusion, the tumor microenvironment model, as previously described, can aid in answering questions related to chemosensitization and cancer relapse. Future testing will be done with patient-derived spine metastases secondary to breast cancer to allow further assessment for combination treatments. This project may lead to the generation of a new combination treatment that can target both cancerous and senescent cells within and surrounding the primary tumor to reduce overall growth, viability, migration, and recurrence of cancer. Thank you so much for listening.
Okay, thank you for the presentation. If we encourage people now to unmute themselves or raise their hands to ask questions. So
If anyone would also like to write the question in the chat box, I'd be happy to answer any questions as the seminar progresses.
All right, I think if there are no further questions, Elish, we can move on to the main speaker.
yeah, and we just want to remind you guys to fill out the evaluation form that has just been posted in the chat by VP Solutions. So moving forward, now is going to be the presentation by Dr. Bartlett, and Dr. Faxmawali will be introducing Dr. Bartlett.
I think there's a question in the chat from Dr. Bartlett.
Yes, there is a question. So the question is, how does inhibiting senescence enhance toxicity of doxycycline?
Hi, I'm not sure I fully understand this question because I don't think it does enhance toxicity. If maybe you could elaborate.
Yeah, you were showing that the size of your spheroids was less when you had the combination of anti-senolytic compounds, didn't you?
Yes, so it's not necessarily toxicity of doxorubicin because doxorubicin can become toxic in the body at too high of a concentration, but it is the accumulative effect, just that it eradicates more of the tumor cells in the model. So senescent drugs, so the senolytics, also target cancerous cells that are senescent, which means that by eradicating them, we also eradicate more of the tumor, is what the model is showing. If that.
You don't prove it shows that. I mean, that's an alternate explanation, right?
I haven't gone into the chemical compounds of it all as of yet.
Okay.
Mm-hmm.
There's another question in the chat. Says, could the use of doxorubicin on senescent cells prevent cancer relapse?
So doxorubicin is a standard chemotherapeutic that is used as of now, where patients still sometimes have relapse. So the idea behind the project that I'm working on as of now is to add senolytics in order to prevent the relapse, because senescent cells specifically will evade the immune response, And so those are the cells that aren't necessarily dividing. They're not being killed off by the chemotherapy. And so they stick around. And so that's what we want to target and eradicate in order for relapse not to happen.
Okay, does not. Yes, so another question came up. This is from Vanessa. She's wondering if you've read about the current hurdles preventing use of senolytic therapy in cancer treatments, and if you have thoughts on it.
Sure. So one of the senolytics that we're looking at is actually FDA approved thus far. And one of the major hurdles was actually that it was thought that senescent cells were actually aiding in cancer treatment because they're cells that aren't dividing. And so one would think that that's good for a cancer. You don't want the cells to divide, but they actually do cause a lot of other problems. So it's more so showing that these drugs can aid instead of do the opposite effect in cancer treatment. Slow and steady work, basically.
So thank you very much, Elaine. I think we're going to move in to Dr. Bailey. And thanks, Eilish. Much appreciated.
And yes, just a little correction. The speaker is indeed Dr. Bailey, not Dr. Berlin. I'm sorry.
I was quickly throwing a talk together in 8 seconds.
Keep you guys on your toes.
Yeah. So Sam, we have the evaluation.
Yes,
Okay.
the link is already popped into the chat.
Okay, great, thanks. So please go to the evaluation. It's anonymous. So thank you very much, everyone, for attending our first seminar. So I would like to introduce Dr. Bailey. He's an assistant professor in the Department of Surgery and Human Genetics here at McGill, and a junior scientist at the Research Institute of the McGill University Health Center. So his overarching goal of his research program is to improve the survival of patients with cancers of the stomach and esophagus. So his group is basically focused on understanding what's the role of coding and non-coding somatic mutations together with alterations to the chromatin landscape in the progression of these cancers to metastasis. So the other thing that his group has been looking at is developing bioinformatic approaches to interpret the function of non-coding genomic alterations to regulatory elements in cancer initiation and progression. So Dr. Bailey, thank you very much. It's all yours.
Okay, great. Can someone allow me to share my screen?
Well, doctors, from your end, under the TV Zoom, you'll see a green icon.
Yeah, it says share screen, but then when I press it, it says host disabled screen share. There we go.
Good.
So you'll have to give me a second. Sorry for this. I just switched to a new computer, so I gotta. Oh. Here, let me move the presentation to my other computer. It might be easier. Come on. Does anyone know how to change the settings on a Mac?
Yeah, which settings are you trying to change?
This is a new computer, so I guess I have to give it
Yeah, you have to give it the permission. So what happens when you accept, you have to restart your Zoom meeting. So you have
okay.
to move out and come back in. Now the other way
Right,
is
so
that,
I'll hang up and then come back.
yeah, and once you just say yes to everything and come back, because the first time you access it, it will ask permission. Or else you could send me the PowerPoints and I could screen share in advance with you.
That might actually work too, but it's probably pretty big. So I'll
Okay.
leave and I'll come back.
Okay, we'll wait for
Is
you.
there, so it'll so I have Zoom open, but what do I have to like? Do you know where it is that I have to give it access?
Well, when you ask share screen, did it pop a menu saying access it, please?
Yeah. Oh, advanced sharing options. There we go.
There you go. Yeah, absolutely. So you have to say yes, basically. Leave the meeting and come back. That should work.
Because everything's blank, which is what it's not. It's
Could be maybe
oh, there we go.
So just distracting people with some stuff, some news in the chat. While
Okay,
Spencer,
let's try this again. Sorry, it's a good thing that I was scheduled for 1:30.
right. Yeah, please open your cam. There we go.
Okay, can you see it now?
Yep. So that was the way. You have to ask permission and then come back. There you go. Amazing. I just sent a little pop-up for a video if you want to open your cam, Dr. Billy.
Yeah.
There you go. Amazing. Thank you.
All right. So sorry for that, a little mishap. It's the first time I've used this computer on Zoom. So I'd like to start by thanking Dr. Mwale for the introduction and also for the organizers for inviting me today to present my work or our work. And this talk is actually brand new, hot off the press. And so I actually haven't given it before. And so it's an opportunity for me to potentially get some feedback on it. So with no further ado, I'll start that. I'm going to change it up a little bit. So since I'm giving this live, feel free to interrupt me at any time. I'll try to keep an eye on the chat or, just speak over me a little bit if you have a question. Find that's easier than waiting till the end. But that's up to you. So I'm going to switch. Everyone can see my cursor too if I move it around, right?
Yes.
Yes.
Okay, great. So I'm going to start this like bucking the trend a little bit, but I've seen this more and more, and I really like it, especially during COVID. So the work I'm going to present today is it's mostly, a very large collaborative effort, and especially with a good friend of mine, Xiaoyang Zheng, who was at the Huntsman Cancer Institute and now he's moved back home to Shanghai and is at Fudan University. But him and a student in his lab, Yan Li, really spearheaded a lot of the functional work that I'll be talking about today. So I do want to give them kudos up front, as I'm primarily computational. So if we move on, now that lead-in talk was very great. Maybe some of you already have a background in this, but since I'm giving it to the surgery department and not like a traditional genomics crowd, I've kind of set up an outline and how I'm going to break down the talk. And so at the beginning, I'm going to give a pretty, brief introduction about precision oncology and how we go about discovering, target genes and cancer driver genes. Very brief. For those of you who've taken, the students in the room that have taken precision oncology, that'll be kind of a bit of a refresher. I'll briefly touch on squamous cancers and SOX2 amplification, and then get into the meat of the talk where we've identified this co-amplified non-coding region in squamous cell carcinomas. And this led to the identification of a specific enhancer that seems to drive the majority of the SOX2 expression. And this also led us to a potential new therapeutic way to target these cancers with SOX2 amplification. So many of you may already know what precision oncology is. It's just a recoining of the term personalized medicine and adding it to cancer. But it's the main idea behind this is to utilize a patient's tumor's genetic makeup or the alterations that are found within an individual's tumor to guide treatment, diagnosis, prevention, et cetera. And this is in contrast to the way things are currently done by most guidelines, where it's a one size fits all. Every patient with a given cancer found at a given location is treated the same way. And since this is surgery, the surgery department, I also want to point out that we can kind of tailor this to many different things and not just like stopping cancer. This can be, since surgery is an effective method showing for curing certain cancers, we can actually tailor this, and that's what a lot of what I'm interested in is to the complications such as metastasis as well as recurrence. So if the surgeons can remove the primary tumor, we can actually hopefully target those specific, like metastasis, which is eventually going to kill the patients, or recurrence of that tumor, and hopefully cure the patient with a combination of both surgery and treatment. So typically what we do in the lab is we'll get a piece of tumor as well as either normal tissue or a blood draw from a patient and perform next generation sequencing to identify both somatic and germline events that could potentially be used to target treatments for those patients. But we're doing this in a research setting and looking for new targets as well as epigenetic or non-coding targets. And so I think the best way to kind of think about precision oncology is actually to use the clinical trial designs to explain it, because that's essentially what we wish to do, right? And so if we think about umbrella trials, that's, this is kind of what most of us think about. And so currently, if each one of these individuals is a cancer patient, and they all have, say, gastric cancer or squamous cancer of the head and neck, let's say, they will have different molecular makeups, but they will have certain genetic alterations in common, which are indicated by these little circles. And so we could target those, group those individuals with the same alterations together and target them individually instead of treating these as one entire entity. And that really lends itself well to this basket trial design. And this, I think, is the ultimate goal of precision oncology, and that's to treat individual cancers based on their genetic profile and not based on the tumor location. So if we look, this is more like, the way that the surgery department is set up into divisions. And this is, all the different surgery departments put together, or divisions put together. So we can treat, the individual cancers with a specific alteration shown here in red as one entity. So how do we identify, cancer driver genes or potential new targets or biomarkers for patient stratification? And so if these are, again, individuals with a given tumor, we profile them for all the mutations or alterations that we can identify. We can map those to genes. I've made all these genes the same size, or cartoon genes the same size on purpose. And then we can map those mutations to those genes. And you can see if we do it this way, and I just want to really point out this, I mean, it's a very simple graph, but I want to really point it out because you'll see a lot of diagrams that look just like it. And so here, this individual here would be this column here. So each column represents an individual patient, each row representing an individual gene. You can see when we present it this way that the green gene has more mutations than we'd expect based on chance. Given the fact that these are all the same size, we can create a background mutation rate in our heads, and we would expect on average 2 mutations randomly in each of the genes, but you can see the green gene has more mutations than we'd expect. And that would be an example of a potential cancer driver gene, or a candidate cancer driver gene in this case. And so here's an example. This is just based on the fact that I have a good example and a visual example to show, but this is an example from melanoma. So again, each column here is an individual tumor sample, and each row is a gene that's been identified as being significantly mutated among this sample of melanomas. And you can see when we present the data this way that BRAF is significantly mutated in over 60% of these tumor cases, making it a potentially ideal candidate for precision oncology because it's found in many people, and we can target it. It appears to be selected for among these cases. And one thing to point out as well is that for the case of BRAF, it's actually tends to be the same mutation, V600E mutation. And so here's the ultimate goal. So here's a patient. This is a PET scan before treatment. This individual has the BRAF V600E mutation. And you can see that the heat map here is showing a high degree of glucose metabolism, and the burden of, you can see the burden of disease in this individual. But following 2 weeks of treatment, you can see that is significantly reduced. And so that's kind of our goal here, is to identify targets that we can that are actionable and that we can hit with small molecules and potentially cure these individuals. So there's more than just single nucleotide variants, but found in cancers, but also copy number alterations or amplifications and deletions. And this is just another visual example. So as I go on, and this is primarily what I'll be talking about today, are copy number gains. And so here's a normal cell. This is Mechan, and you can see you have 2 copies. This is fish. So you're labeling both copies of Mechan. This one's from, say, your father, and this one was inherited from your mother. But you can see within the cancer cells, you have many copies, like hundreds of copies of this specific amplification. And when you look at the cell, I just want to point out that many of these gained copies are actually outside of the chromosomes, and so they're extra chromosomal. And recently, this is kind of just an aside, these have been shown to be actually circular, very similar to plasmids, extra chromosomal DNA. But we'll present this data very similar to the way we presented the single nucleotide variants. And here we're actually combining the two. So this is an example of Mechan amplification in medulloblastoma. It's actually not that recurrent among medulloblastomas, but you can see that each individual is the column and each gene is the row. And one thing I just want to point out, that there's many patients where we don't know what the driver is. So if you look over here on the right of these subgroups, that they're quite blank and quite sparse. And with the exception of a few of these driver genes, there's not much, not many targets in many tumors. So this is the last kind of cartoon to set up the rest of the talk. And this is just another way that we tend to present these copy number gains. And so if we typically draw them as red bars or a red heat map, and the length of the bar and the genes around them is what the region that we see that's amplified. But remember, we don't necessarily know where that gain is. It could be anywhere in the genome. It could be inserted into a neighboring chromosome. It could be a tandem duplication. It could be on extracellular DNA, similar to the image I just showed you before. But what we're looking for, again, is recurrence. So we're looking for, the same region being recurrently gained or amplified across multiple patient tumor cells. And then that will nominate genes within that region as potential oncogenes, because the gain in copies is thought to lead to a gain in expression of an oncogene. And then we can play games with this data as well. And so if you're fortunate enough to identify tumors with smaller gains or more focal gains, you can actually whittle down the candidates to maybe just this purple gene as being your potential candidate oncogene in that region. So just to move on to squamous cancers. So squamous cancers represent one of the most frequent, if not the most frequent, solid tumors. And they can occur in diverse tissues. Now, I'm mostly interested in those that affect the esophagus as well as the lung, but I mean, head and neck and cervix are also important. And the idea, if you remember the basket trial, is to potentially treat all these cancers in the same way. And so they're known to actually have an amplification or gain of SOX2, which is a member of the SRY box transcription factor family. And it has a prominent role in pluripotency. It's actually a Yamanaka factor. So the addition or the expression of SOX2 along with NANOG and KLF4 will actually convert a fully differentiated cell to a stem cell. And then it's also essential for maintaining that, because it's essential for maintaining the self-renewal ability of basal cells, which are thought to be the cell of origin of squamous cancers. And so this is just to illustrate, the same thing occurs within these squamous cancers. This is lung squamous cancers, but you can see now that everyone knows how to read these plots that 56% of lung squamous cancers harbor a gain of SOX2. What I'd like to point out here is that you see that these tumors also have gain in PIK3CA, which is a known cancer gene, as well as TP63. And you can see that they tend to co-occur. This gain tends to co-occur across all these tumor samples. And that's because what's typical of squamous cancers is that they actually gain the entire long arm of chromosome 3 and not just the region around SOX2. And so that's why we have that. But if we look, actually, let's leave that there. So that's the reason for that. But there's evidence to show that SOX2 appears to be one of the primary or the primary hit in the region. However, these other ones are likely also playing a role in these cancers. And so this is just to kind of further point to the reason for focusing on SOX2, at least for this project, is that it seems to be associated with survival of cancer patients, although there are some conflicting results. For example, in the lung squamous carcinomas that I just mentioned, the increase in SOX2 expression has actually been associated with a favorable prognosis, and there's some conflicting results in gastric cancer. But given its role as a stem cell factor, I believe that overall it's going to be involved in recurrence and the cancer stem cell hypothesis and dormancy. So just one more intro slide for the rest of this is that our view of these gains has been changing more recently and evolving, because our initial analysis were typically focused primarily on genes, and we talk about the oncogenes and tumor suppressors. However, these gained regions tend to include regulatory elements, insulator elements, many different functional non-coding elements, as well as non-coding genes. And so there's been a goal or a change or shift in the field to focusing on not just genes, but what else is being amplified or gained and rearranged in these tumors. And there's been several examples of regions that are gained that do not contain any genes. They tend to contain either super enhancers or regulatory regions. And there also was a recent publication, which I'll get to, where not only are genes appearing to be selected for, but also these regulatory regions appear to be selected for, even though the gene is amplified. And that's more in line with the story that I'm going to talk about today. But this suggests different ways of activating oncogenes, as well as potentially new therapeutic targets. Everyone's okay so far? Okay, great. So this is one example. This is actually from Xiaoyang. This is work that he did in his postdoc. where he discovered focally amplified super enhancers nearby known cancer genes in epithelial cancer. So for example, here you can see that there's recurrent gains in head and neck cancers or squamous cell carcinomas that are just upstream of KLF5, but do not include KLF5. And he goes on to show that this is an important enhancer region. The same is true also for MYC, another well-studied oncogene. There's different enhancer regions appear to be focally amplified in these tumors, in different tumors, in this case, lung adenocarcinoma or endometrial carcinoma. And that kind of mimics the enhancer or regulatory landscape of those tumors. And this is the paper that I kind of just focus on a little bit. It's work by Peter Scaccieri and my former mentor, Matthew Lupian, where they show that also in these cancers, and this is actually those ecDNAs or circular amplifications or genes that get circular like plasmids, and they show that these amplifications also tend to include enhancer elements. And so there's a shift. If you can see, this one's probably maybe not the cleanest example, but this is EGFR, and you can see that overall these are many different patient samples. You can see that there's a slight shift towards this green region that tends to include enhancer elements together with the EGFR gene among many different tumors. So going back to squamous cancers and SOX2, we reanalyzed the TCGA, so the cancer genome atlas sample. So there's about 1,500 samples that were profiled using SNP arrays. And we ran a logistic analysis or analysis for identifying recurrent copy number gains and losses, but in this case, I'm only showing the gains. And we can see that the most predominant region is the SOX2 region on chromosome 3. I just want to point out here that in gray here, this is all the amplification. So this window here represents the entire Q arm of chromosome 3. And then for the remainder of this talk, I'm going to focus primarily on these ones that are shown in blue, which are what we term focal amplifications. And so we define them as being anything less than 10 megabases, where we had a continuous segment, including the SOX2 gene. However, you can see that there are some of them that still have gain of the chromosome or the chromosome arm as well. But what was interesting is when we looked at these, and so this is pan squamous, and we looked at the focal amplifications, you can see that this black line here indicates where SOX2 is located on the most recurrently amplified. Remember that little cartoon where we're looking at all the focal amplifications? You can kind of whittle down the genes. There are a few other genes in this peak, but SOX2 is located just on the border of this gained region. So we decided to look at other cancers that also amplify SOX2, in this case, gliomas. And so we had about 1,100 gliomas available through the TCGA data set. And you can see that we also identify SOX2 as being significantly amplified among these cancers as well. However, it's not nearly as frequent among gliomas as it is among squamous cancers. But you can see that there is a shift in the other direction. So this time at 5' or to the left of the SOX2 gene when compared to the squamous cancer peak. And so if we zoom in on this a little bit, you can see that the crossover point between these two amplified regions occurs right at SOX2. However, there appears to be significant or selection for the neighboring regions, 1 3' for squamous cancers and 1 5' for the glioma samples. So how do we go about potentially testing for, whether this is a significant event or whether it's just a random occurrence? And so there's no real method currently available to do this. Morton et al. Provided a method, so we adapted their method. They didn't. It's actually not published, but their analytical strategy was essentially the same as what I'm going to propose here. And so we'll go through what we came up with. So if we assume this little cartoon represents the shift or the skewed SOX2 amplifications, and that there's a non-coding region indicated by this peak here in green, that's also selected for together with the SOX2 gene, we can take those segments or amplicons, and we can randomly shuffle them so that we keep the lengths the same. So each patient's length of their fragment is the same size, but we make sure that it can only slide to, but it always has to include the SOX2 gene. So we're going to create our own null hypothesis here of a random, what would be a random occurrence. And so we'll do this 1,000 times, and we'll just shuffle these around. And then we get kind of like our null distribution of what we would expect if this was a random event. And that's indicated here in green. So this stepwise thing is that we've binned the genome into 1,000 base pair bins. But the what we observe for the actual observation is in red, and then the null hypothesis, so to speak, is in gray. And you can see that there's a significant skew to the right. So this whole thing is significant. And right, this is what we'll be focusing on, is this green area, is where 98% of these highly amplified SOX2 have a gain of this region as well. It also could be that these guys have it as well. And I'll just show that we're using actual breakpoints of the segments from the analysis. But you can see, like, for example, this individual where you have a gap. And so this section would not necessarily be included in that analysis. And so it's difficult to come up with a heuristic to do that from this style of data. But you can see there's a significant enrichment of a region in this region. Actually, there's a shelf, and you can actually see that visually by eye. And you almost see that there's a significantly less amplification on this side, indicated by the white bars that you can start to see creeping in here. So what is in this region? What can we hypothesize in this region? And this is just a little background for epigenetics for those of you that may not be up on this type of data. So the DNA is wrapped around nucleosomes, and so those nucleosomes are made up of histones, and those histones have tails that protrude out from the chromatin, and they can be post-translationally modified. This is similar to serine phosphorylation for enzymes, to activate enzymes or other proteins. And so when they are acetylated, they tend to be active, and that creates permissive chromosome or chromatin or open chromatin. And so open chromatin is where the transcription factors and transcriptional machinery can actually bind to the DNA, and it's accessible. Methylation tends to be the opposite, when that leads to a condensed chromatin, where the transcription factors cannot bind there. So we have several different tools or genomics tools that we can use, or assays that we can use to identify these regions. So one of them that I'll be talking about next is a TAC-seq. Essentially, we're using a transposase. That transposase, wherever it can insert into the chromatin, is likely open chromatin. So we sequence those regions, and we'll get peaks. And those peaks are all the potential regulatory elements, including insulator elements, present in the genome. And then we'll also utilize ChIP-seq to actually pull down acetylated histones that can mark active promoters and enhancers. And so the TCGA is also profiled relatively recently, more than 400 different tumors across different tumor or cancer types using a TAC-seq. And so we can leverage that data as well. And so when we look at the two regions that we identified, the one in glioma and the one in squamous cancers, you can see right off the bat. So these peaks are actually the average signal intensity across all the tumors that were profiled for that given cancer. So these are all the squamous cancers separated by type, and these are all the gliomas. This is low-grade glioma and glioblastoma. And you can see quite clear that they're very similar. And so this is about treating these cancers as the same in our pan-cancer analysis. But what you can see in blue here is that the squamous cancers have enhancers or regulatory elements that are within the selected region found in squamous cancers, and the opposite is true for the gliomas. You can see that there's lots of regulatory elements to the right of the SOX2 peak, or within the SOX2 peak, but to the right, and then in squamous, the reverse is also true. And so we extended this to to cell lines so that we could actually functionally validate these enhancers. And so when we look at cell lines, and now this is ChIP-seq data, so these are active enhancers and promoters that are marked by histone 3 lysine 27 acetylation. But the peaks are quite similar and in the same location. And we're going to be focusing on 8 enhancers that we identified that were, so these are SOX2 high expressing cancer cell lines or tumors, and these are tumors that do not express SOX2. And so we identified 8 enhancers that were common to these ones, but not common or not found in the non-expressing SOX2 squamous cancers as well. One interesting thing to note is that these neighborhoods, these are we'll get to this in a few minutes, but these neighborhoods are chromatin interactions, and there appears to be 2 kind of neighborhoods here, active regions, and this one seems to be active in gliomas, and this one in squamous cancers. We also had data available for 112 whole genome sequencing samples, also through TCGA. We performed logistic analysis just using the segmentation files. Again, as expected, SOX2 is recurrently amplified among these tumors as well. But the whole genome sequencing data also allowed us to identify structural variants, so duplications, inversions, translocations, and deletions. And what we found was that some of the tumors that also didn't have a noticeable gain in this region also had tandem duplications in the regions that included just the this enhancer cluster or super enhancer near the SOX2 gene, and this is really reminiscent of the lineage specific amplifications that I mentioned previously. Again, pointing towards this region being somehow important for these squamous cancers. And so the next question is, so now we have the gene SOX2 that we believe to be a cancer driver, and we have enhancers, but what the next piece of evidence we need is whether these enhancers actually do in fact regulate SOX2. And as I alluded to, although we think of DNA as like a linear molecule, it's actually organized in 3 dimensions in the nucleus. And so we also have multiple genomics technologies now where we can assay which regulatory elements are in contact physically with which promoters, et cetera. They're all based off this method, and I won't go through it unless you guys have questions about it, or you can ask me offline later. But they're all variants of this 3C or chromatin conformation capture technology. And in our case, we're using high ChIP. So essentially, we're enriching using the same antibody as ChIP, the 27 HVK27AC antibody, to pull down interactions that involve active promoters and enhancer elements. So when we perform these assays in the cell lines, so these are the 4 cell lines that I showed you earlier that have high expression of SOX2. You can see that we get very strong interactions between the SOX2 promoter and these enhancer elements. Specifically going to focus on this E1 to E3 enhancer region, but that's present in all these SOX2 high expressing squamous cancer cell lines. We did one cell line where there was no expression of SOX2, and we found no significant loops in that case. So now to test whether these enhancers are functional or whether they're important in driving SOX2 expression, we turn to CRISPR interference and CRISPR activation. The reason for not just using traditional CRISPR is that there is a toxicity in highly amplified regions. So the number of cuts that the CRISPR would cause also causes cell death for those cells. So there's like a increased false positive rate among CRISPR screens in regions that have high level amplification, which you can adjust for, but we chose to use CRISPR-I and CRISPR-A. So instead of the Cas9 creating a double strand break, the Cas9 is actually inactive or dead, and it's fused to a repressor in this case. And so when we target it, we use the same sgRNA guides, and we target it to an individual enhancer that will repress that enhancer element, and the opposite is true for the CRISPR-A. So instead of being fused to a repressor, it's fused to an activator, so we can activate that enhancer or promoter. So when we perform these studies, or when Xiaoyang and Yan Li performed these studies, they tested all 8 enhancers, but here we're only showing the first 5. We saw that only the E1 enhancer seemed to be a predominant enhancer driving the expression of SOX2. So this is the change in SOX2 expression using the sgRNA guides. And so the range was between 60 and 82% reduction in SOX2 expression for just for the E1 enhancer. And this was consistent across all the cell lines tested. And it occurred for both, RNA expression as well as protein expression. And furthermore, when we performed RNA-seq on a cell line where we had knocked down or inhibited the E1 enhancer, and we can compare those profiles to cells that have been knocked out for SOX2, and you can see that we get a negative enrichment for SOX2 activated genes and a positive enrichment for SOX2 repressed genes, as one would expect. And furthermore, when we take that and we correlate the gene expression with those tumors in the TCGA RNA-seq data set, it correlates well with SOX2 expression in both the activated and repressed genes. So now can we do the reverse, right? So we take those cell lines that I mentioned previously that do not express SOX2. Can we activate SOX2 expression by targeting the coactivator to the E1 enhancer? And the answer is yes. So here in each case is 3 different cell lines. So you could view this as 3 different tumors. If we target the E1 enhancer to activate it, we see an increase in expression at the RNA level as well as the protein level in those tumors. And then when we perform RNA-seq on those samples, probably as expected, SOX2 is the most changed gene in that region. And then we did the same kind of experiment. So we see a positive enrichment in this case with the SOX2 activated genes and a negative one with the SOX2. This is gene set enrichment analysis, if I forgot to mention that earlier, with the SOX2 repressed genes as expected. So that E1 enhancer seems to be a dominant enhancer for regulating SOX2. And so I guess this leads to the final question, at least for this part, was, if we modulate this enhancer, can we affect, cancer phenotypes, through its role regulating SOX2? And so the answer is yes. So in this case here, we're targeting the sgRNAs to the E1 enhancer again, and we see a reduction in cell proliferation, or over 50% reduction in cell number across 3 different cell lines here. And then we also see this a reduction in tumor volume in a xenograft model of one of those cell lines. So can we now knowing this, that there's a dependency of these cancers on an enhancer as well as SOX2 because they're co-amplified and co-selected for, can we come up with a potential therapeutic strategy for these tumors? And so one approach that we used was potentially to use BRD4 inhibition. So BRD4 is a co-activator that's recruited to enhancers by transcription factors and to drive expression. So can we disrupt this kind of interaction here and inhibit the expression of these oncogenes? And this has been shown for other genes such as MYC. So first, we tested whether BRD4 was in fact found at these enhancer elements, as well as whether the E1 enhancer inhibition could actually affect BRD4 binding. So instead of histone marks or a TAC-seq here, this is actually CHIP4 BRD4 itself. And so each one of these peaks are where BRD4 is located on the genome. And so here's our enhancer set, and this is the SOX2 gene. And you can see when we inhibit the E1 enhancer that we have a significant reduction in BRD4 at this enhancer cluster, the entire enhancer cluster, not just E1, and at SOX2 as well. And if we look at all SOX2 regulated or bound regions, or those found with a motif, we see about a 30% reduction in BRD4 binding. Now remember, other transcription factors that are also binding can also recruit BRD4, not just SOX2. And this was consistent across multiple cell lines that we were using. And so here's the strategy. Can we again inhibit BRD4 to affect SOX2 by disrupting this interaction between the enhancer elements and the promoter of SOX2 using BET inhibition? And then I put this here to remind myself whether we got some interesting data from this. And so whether this looping model, whether disrupting BRD4 actually changes the chromatin architecture is interesting, because we have data that this model may not be correct. Not the fact that inhibiting BRD4 won't affect expression, but the fact that that the chromatin architecture will be impacted. And so to do this, we are using a PROTAC or a proteolysis targeting chimera molecule. This is ARV771. And so just in a nutshell, this PROTAC targets a protein of interest, in this case BRD4, and it will recruit the E3 ubiquitin ligase to it to ubiquitinate the protein of interest and cause it to be degraded by the proteasome. And so again, if we treat these cell lines with the BRD4 inhibitor, you can see we see a dramatic reduction in BRD4 at these regulatory elements. However, we do not see the same reduction in 27 or H3K27 acetylation. So that's still present, but you can see that the number one enhancer in this region that's affected is that E1 enhancer region. And furthermore, when we look at gene expression, we can see that SOX2 is one of the most highly impacted genes after BRD4 inhibition, likely due to the number of copies of SOX2 and the number of copies of this enhancer in that region. You can see after 6 hours, there's almost little to no detectable SOX2 in these cells. And this was the interesting point that I kind of alluded to. So we actually originally thought that this would also impact the chromatin looping at this site, but it appears that BRD4 inhibition, there's no significant change in the chromatin architecture at this locus. So it seems to be that that although it's important for transcription, it's not necessary for maintaining the chromatin loops. And then this was recently reported just this year in Nature Com. So, I believe this is probably true that BRD4 inhibition is not required, or even though it disrupts transcription, it's not required for these promoter enhancer contacts. But what is true is that here is the opposite experiment, and then now we're using the CRISPR-A to activate that enhancer. The activation of that enhancer is enough to actually have that chromatin architecture rebuilt. So, inhibiting BRD4 doesn't have an impact, but if we activate that enhancer element, the chromatin architecture will be established in that region. So that was an interesting finding. To summarize what I said today, squamous cancers and gliomas selectively amplify enhancer regions, both 3' and 5' in their respective cancers. This is likely caused by the underlying, chromatin architecture that's found in the cell of origins of those cancers, but we don't have direct proof of that. We reveal that there's these distal enhancers are looped to the promoter of SOX2, and that there's a predominant enhancer, even though there's an enhancer cluster in this region. There's one specific enhancer that seems to be driving SOX2 expression. Furthermore, although SOX2 itself is not targetable, the fact that these tumors are reliant also on the regulatory architecture, and that they're co-amplifying those regulatory elements, this points to a potential vulnerability that we can assess, and we give an example of this using the PROTAC-mediated BRD4 degradation. So there's a potential strategy here for treating these tumors. Remember, SOX2 amplification represents 60% of squamous cancers. So this would actually be a good, biomarker for these tumors to use and potentially treat these. However, this will require further preclinical evaluation and animal models, though it's suggestive at this point. And then finally, there's just that interesting piece of data there in that recent publication is that, enhancer activation seems to be required for initiating enhancer promoter contacts. And this is more basic science level. But the BRD4 degradation doesn't, although it has a large impact on transcription, doesn't appear to have any effect on chromatin looping. So with that, I would just like to end. Hopefully we're on time because I don't have a clock, but and then I'd like to thank once again, thank all my collaborators, especially during COVID. They really spearheaded a lot of the functional work due to differences in how different institutes respond as well. So I can't thank them enough. And thanks for everyone for staying around and listening.
So thank you very much, Dr. Bailey. I liked the introduction, really excellent introduction. So any questions, please feel free to write in the chat or just unmute and ask.
Can I just pose kind of a neophyte type of question in this, that looking at the SOX2, that it has so many roles apparently that we from going into the pluripotent stem cells, and then you go in the opposite thing, you go oncogenesis. So it looks like it requires an extremely refined balance on how much of it we should have in a spatial, in a specific locus or in a specific situation. Another thing that I've noticed is that I like very much the way that you did the serialized experiments, so I can see that actually as SOX2 expression, it's kind of increased in various type of variables. And another thing is there are a lot of variables in play. So beside the histones that go, that there's a lot of scaffold proteins that are around the DNA, and because of the three-dimensionality of it, I think maybe my question is, do you think that the steric type of configuration could be considered also as an angle to look in this type of research? Thank you for the presentation. I'm seeing the SOX2 right now in a completely different light, and it's obviously way more complex than I initially thought as a oncogen. And I would really like to hear, what do you think about this three-dimensionality and kind of this kind of yin-yang of where is the balance?
Okay, so I'll try and answer that. You tell me if I hit on all the points. So I think here, so BET inhibitors have been around for a while, or BRD4 type inhibitors like JQ1 have been around for a while, and they're very toxic. And so I guess the way to think about this, the reason why I think this worked is not so much the chromatin architecture. Now, the chromatin architecture is important here, but I think a lot of what we're seeing is potentially these ecDNAs, so these circularized gains. And so if you think about having lots of copies, I think the reason why the BET inhibition works and it seems to target SOX2 the most in these cancers is because you have many, many copies of that enhancer as well as SOX2, right? So like if you think of a regular genome, if you were to hit it with BRD4 inhibition, you would affect most of the genes, if not all of them, right, that are being actively transcribed. Right? But then the reason why this is seems to be specifically targeted, though it's not, right? Because this is like a global, like BRD4 is recruited all over the place. It appears to be more specific because of the level of expression, right? So I have a feeling that this extends to also the MYC amplifications, the Mechan amplifications I mentioned, EGFR potentially, that those cancers, so highly amplified cancers with known driving oncogenes would probably be the best bet for these BRD4 inhibitors in my mind, at least what I've seen currently. So yes, the architecture is important, but in this case, I think it's because the architecture is circular or whatever, or like extra chromosomal, if that makes sense. So some of what we're seeing, remember, we can't currently really separate between where those loci are, right? So those close proximity analysis could be because they're sitting on, you know, a potent, like kind of like a plasmid, you know, a cancer generated plasmid, if you will, or if you let me go there, but that's kind of how I view it. I hope that answered your question.
Yes, yes, and yes, it does. This is a really very entangled SOX2. It's a really entangled thing, and I think that going through the BRD, it's a good path to very specific treatment of cancer for specific cases.
Yeah, and so just to touch, sorry, I just remembered your other point. Like, so just to touch on the relative amount, I do think that's true, right? So like,
yep.
So remember, SOX2 is expressed in these progenitor cells, right? And so the amplification, now how it occurs or when it occurs or why it occurs, who knows? But it is like it's already expressed, right? That's kind of what we're hinting at with the looping structure is somehow playing a role in the gain as well, right? So if you look at the chromatin architecture, it kind of mimics the gain. So I don't want to come out and say it, but like, you know, there's something to the original chromatin architecture and the gain, and that it was already expressed to begin with, right? Because those loops and connections were already there at the beginning, and then that was gained. So I don't know about the relative amounts, but it's keeping that expressed. So that's a, you know, there's a stem cell factor, but the cancer, you know, it was expressed in the initial cell, and then these gains are allowing it to continue to be expressed, right? So those basal cells have a self-renewal capacity because of SOX2. As they become cancer, they have maintained that ability. So you could look at like the same thing as with telomerase promoter mutations, right? That's a way to make sure you live forever, right? Like they get a mutation in their promoter and they don't worry about their telomeres shortening anymore, right? So you know, that's if you humanize or humanomorphize them in a way, if that makes sense.
Thank you very much. Yeah. I like it was a very good very visual explanation, and I like that you go into uncharted territories.
Yeah, I'm mostly focused on non-coding. It's hard to present sometimes because you got to like give them the background, but no, we're mostly interested in non-genic regions. So we have a big focus on non-coding mutations. I'm actually that telomerase promoter mutation is actually one of the most recurrent events in cancer, and was only found in 2000, you know, I think 13. So there's hopefully a lot more to be discovered. I don't think we'll find another one like that, but it's the most recurrent, if not the most recurrent, overall the protein coding mutations that have been found today.
So do you get to look at the interplay with TP63?
So I didn't, not for this gene, I haven't really paid attention. P53, you know, I typically assume that it's affected in all of them. There's like 70% or 75% always have a P53 mutation or loss, but I haven't looked at specifically at the interplay with it, no.
Okay. So what about with SOX1? Because SOX1 is supposed to do the opposite. Do you see any decrease in SOX1 in your patients?
So I haven't specifically looked at this. In this case, we were following the genomics, right? So we were following the fact that SOX2 was present. So we haven't went into, you know, the specifics of other downstream regulators. We don't see a SOX2 gain, let's say, or loss. It doesn't ring a bell as being one of the genes brought up as we're currently affected. But yeah, I would be speculating at best, about that.
Yeah, that's really interesting. So any other questions in the chat? We still have some time. Any questions from the audience?
Let's see if I can poke someone.
So with this type of cancers, when especially on the skin, there's the sun factor, the UV factor. So
Yeah.
how does that? What has that got to do with SOX2?
So for that case, I don't think that one's really impacting SOX2, because that's those would lead to point mutations. So this might, you might have, it's like kind of like a substrate mutagenesis. You can think about it, right? And so that over time you accumulate these mutations, or you gain, you probably not necessarily accumulate, but you gain these mutations, and some of them become selected for, right? So some of these point mutations caused by sunlight, I forget, it's thymine dimers, but I'm completely drawing a blank on the actual mechanism. But so I think it's a C to T transition or something like that. But so this would be like the substrate for selection on those cancers, right? And so some of those might be your P53 mutation or so forth, right? Do you understand? Does that make sense? Yeah. But for this specific SOX2, so if you remember when I presented, like there's very few, if no, mutations in SOX2. It's completely like a copy number driven, and the exact mechanism for these copy number, like what causes like the these gains is, I'm not 100% sure.
Yeah, it's really fascinating. Anything we can do to suppress the overexpression of SOX2?
Well, that was the hopefully the main take home is that we may be able to treat them, because we can't target SOX2 itself. That if we treat them with these BET inhibitors or these BRD4 inhibitors or something similar, right? So now we know they're dependent on the transcriptional machinery. It's not just like an amplified gene, so to speak, or an amplified gene in its promoter. They actually require that enhancer element to also be co-amplified with it. So now that that's also shown to be required, like, it's not just the gene that needs to be amplified, but you also need that regulatory structure or those regulatory elements together with it. We can start thinking about inhibiting the transcriptional machinery in those cells. The issue there is that, and that's kind of what I was trying to get at with the number of copies, is that there might be and maybe this is the kind of the comment that was given, there might be a balance there, whereas like too much would be toxic because your cells still use the regulatory machinery, right? Or the transcriptional machinery. But in these cases, it's highly amplified. So you may be able to kind of find a sweet spot. But I'm speculating. We'll see in future studies whether, this will work in mice or so forth, right? So.
Yeah, it's a really interesting work, really wonderful. And thank you very much. If there are no other questions, I would like to thank everyone for attending this first seminar, and we'll be having the seminars every Tuesday, every week. So thank you very much, and we'll be having the students as well. And don't forget to put your evaluation in the chat. So thank you, everyone, and thank you, Dr. Belli, for that wonderful and exciting talk.
Thank you. Thanks for having me.
Thank you.
Bye.
Bye.